I'll hand it over to Professor Sadamani from the National Center for Biological Sciences to welcome everybody.
Thanks, Pavitra. Good morning, everyone. Thanks to the communications team and NCVS. Outside In is a series of talks that we have been hosting, and it's in the topic of ecology, and it is by various experts in the field. It is also an initiative that was taken up during the lockdown to get the high school students, children, and young adults a glimpse into what goes on inside the laboratories. Yeah, so this week's theme is on plants, and I am very happy to have Dr. Radhika Venkatesan, an expert herself in this area, to co-host this series of talks. And today we have the last of the three. And I would request Radhika to introduce the speaker for today, please. Radhika. Thanks.
Thank you, Dr. Saudamini. So welcome all to this Outside In series focusing on plant sciences. So today we have Dr. Jyothilakshmi from National Institute of Plant Genome Research in Delhi. She's been working as a scientist in NIPGR since 2014. Dr. Jyoti obtained her PhD from Friedrich University in Jena, and after a short postdoc at Cornell and Max Planck Institute for Chemical Ecology in Germany, she has been running her own lab in Delhi. She is a recipient of several awards, including the EMBO Global Investigator, and she also heads the Max Planck India Partner Group. Her major research interests include understanding molecular interactions between plants and insect herbivores, as well as plants and microbes. I think today she is going to tell us all about plant-microbe interactions. And now I hand over to Dr. Jyoti to give her seminar, and I'm very happy that she kindly agreed to give a seminar in these difficult times. Thank you, Jyoti, for joining us.
Thank you, Radhika, for the kind introduction. And for this opportunity for the talk. I'll start with the slides without any further delay. Just a moment. Can you see the slides?
Yes.
So good morning, everyone. I'm Jyoti, and this is my institute where I work in, the National Institute of Plant Genome Research in New Delhi. And the title of today's talk is on plants and their microbial friends, and what determines this balanced alliance that a plant root has to, where a plant root has to interact with a good microbe. So now if you look at this picture, you can see a lot of green plants all around. And not all the microbes in nature are bad for plants or humans. So some are also part of a good, giving us a lot of advantages. And through this talk, we are going to see examples of microbial alliances which are good for the plants, how they are good, and how is it that a plant is able to recognize these microbes as being good and using it for its advantage. This is what I'm trying to tell through this talk. So we'll call them microbial superheroes because they are nothing less than superheroes. You don't see them. They are mostly in the rhizosphere of plants. And they have a lot of effects which help plants to sometimes promote their growth, they improve stress tolerance, they enhance nutrient uptake. So these beneficial interactions are very important for plant fitness of roots with these beneficial symbiotic microorganisms. And they're naturally present in the root rhizosphere. And majority of these interactions result in enhanced nutrient acquisition, which is important for the plant. So before I'll just give you a brief outline of how I wanted to give the talk. I will give you two examples of which are very well-known examples of a good plant microbe interactions, which are seen in majority of land plants. And then in the end, we'll come to the system that I'm working on, and just a few results. We'll try to see how they communicate in such an interaction process and how different it is from a plant pathogenic interaction. Now, plants form these intimate symbiosis, and two very good examples you might have already known before is the nitrogen-fixing bacteria and the mycorrhizal fungi. Now, mycorrhizal fungi, or the AM symbiosis, as we call it, is an extensive fungal, a fungal network on the roots of the plant, which facilitates nutrient uptake. Now, this is present in majority of land plants, and it's an ancient type of symbiosis. There is also some other form of way how they form symbiosis, for example with nitrogen fixing bacteria, which live in the root nodules and fixes nitrogen, which also enhances the nutrient capability of plant. And these symbiosis have evolved almost with the plant evolution. Now let's start with the Arbuscular mycorrhizal fungi, and they associate with almost 80% of land plants. So if you look at how old they were in their evolution, you can see that around 400 million years ago, these associations are already known. So they are as old as land plant evolution. And the way they communicate has also evolved over time, and we are trying to see now how is it that they communicate with each other. Now, mycorrhizal fungi is a fungi that trades soil nutrients for carbon. So there are two kinds of them: an arbuscular mycorrhizal fungi. These are the fungi which form something called an arbuscules inside the root host, and an ectomycorrhiza, which associate only with few plants. Now, why do plants need this association between AM fungi and plant roots? So plant roots as such have great difficulty taking up phosphate. Phosphorus is a nutrition element for the plant, and they cannot take up insoluble phosphate in very efficient manner. So the way their root growth is, if these yellow dots are phosphorus spread across the soil, and this is a root growing, they have limitations in how they can uptake this inorganic phosphate. They cannot reach all through the soil. Their root hairs do not reach everywhere. And so there is a depletion zone beyond which they cannot uptake phosphorus. So whatever was near this root hair, the root hair has tried to absorb it. But beyond that length of the root hair, it is not able to absorb. And this is where such microbial interaction helps. For example, in AM symbiosis, you see this blue lines. This is the hyphae of the mycorrhiza, which extends further into the soil and greatly enhancing this phosphate assimilation. So it gives that root an extra surface area which it can absorb all the nutrients other than what its root hairs can do. Now, what happens in the plant cell when an AM fungi penetrate into it? So the fungi produce what is called an arbuscules. This is an area where they exchange all these phosphate which is taken up from the soil. And the fungus, what does the fungus get in exchange? The fungus gets sugars produced by photosynthesis from the plant, and the fungus provides phosphorus from the soil. And this is the site where this exchange between the phosphorus derived from the soil and the plant take place. In fact, 20% of photosynthetic carbon is directed towards AM fungi. So this interaction is so useful to the plants that they have tried to partition also their carbon, share to the AM fungi development. Now, how do they, this is what I majorly want to tell you through this talk, how do the plant root and a symbiotic organism know that they have to that they are good? How does a plant root know that there are so many microbes in the rhizosphere? So how does the plant root know that this is a good microbe? Or how does the microbe know that this is a plant of my interest, I can go and colonize it? So for that, a chemical dialogue is essential in most of the symbiotic interactions, and they are very well-known examples. So for example, in AM symbiosis, it's very well known that the plant root produces strigolactones. These are a kind of plant hormones, and also which initiates fungal branching, hyphal branching. So the plant produces this small metabolite and releases it into the rhizosphere. It comes out as a root exudate. And when this fungus gets the signal that there is a strigolactone being released by these specific roots, it in turn activates the fungi to release another small molecule which tells, hey, I'm your friend. So he releases something called a MIC factor here. I'll come into the chemical structure of the MIC factor, but just keep in mind this is the chemicals which are produced by the fungi to make itself recognized as a symbiont. Once this communication or dialogues happens between the root and the fungi, the plant permits it to penetrate its cells. Yeah, so this is critical for this chemical dialogue is critical for the initiation of AM symbiosis, and it happens in almost a majority of land plants. Now, what is known about the structure of these make factors that we are talking about? The identity of make factors is very recent. It is actually the nodulation factors which were first identified. The make factors were identified in 2011, and they are actually lipocyto-oligosaccharides, or they are chitin fragments with decorations, if I put it simply. So chitin is present in the cell wall of fungi, and these are polysaccharides. So this is the chitin moiety here. They are acetylglucosamine sugar residues, which you see here. And this MIC factor has also a lipid moiety at the non-reducing end, and there is also a sulfation at the reducing end. So this is the basic structure of how a lipocyto-oligosaccharide is, which is called as a MIC factor here. Now, this is not only the only factor which is responsible for the plant recognizing the fungi as a good one. There are also something called a short chain oligosaccharides, which do not have these decorations. For example, here you have a fatty acid attached to this, or a lipid attached to this chitin fragment. So sometimes some MIC factors are also without these decorations, and they can be chitin 4 or 5, CO4 and CO5 polysaccharide, which also acts as MIC factor. So MIC factors are two things, and each host, that is each kind of different rice or different other plants, produce their own specific MIC factors. So there will be variation in the number of these chitin moieties. There will be differences in the fatty acid composition. So each host will produce its own kind of factors which are specific to it. Now, what happens once such an interaction occurs? The plant has given its trigolactones, and the fungus have told, see, I'm MIC factor, and now we are friends. So what happens is that this communication results in the AM fungi being able to colonize these plant roots. Because if you can remember the earlier slide, they should form an Arbuscule. So this is something, a kind of a structure inside the plant cell which the fungi has to form. So the communication allows this colonization to occur. And one of the first steps after this colonization, what you see when a MIC factor is being recognized is initiation of these calcium spikes in the nucleus. So the cells understand that the node factor is released and the fungi is ready to grow inside it by sending out these nuclear transients in its nucleus, calcium transient in the nucleus, and the plant recognizes it as a good fungi. Now, these calcium transient can occur also due to pathogenic fungi, but the kinds of transient that occur are different for symbiotic fungi and for pathogenic fungi. They also occur in different organelles here. So the size and the shape of this calcium transient tells a plant, yes, now you can move forward and colonize. You seem to be a good person for me. Yeah, so this is some in brief about what is known about AM symbiosis and its chemical communication. Now we'll come to the next symbiotic interaction, the nitrogen fixing bacteria and legumes, which is a more modern kind of an interaction. So why are we talking about it? So when you talk about nitrogen fixation, it's important to understand that nitrogen is abundant, but it is unavailable. So nitrogen must be fixed or reduced. It's present with this triple bond. And so it has to be broken to be made biologically active. So conversion of this nitrogen to ammonia is what makes it biologically available to the plants. And plants do this by using nitrogen fixing bacteria. So the root nodules and the nitrogen fixing bacteria that grow inside such root nodules ensure that plants get enough nitrogen supply. But this happens only in specific plants like leguminous plants. And the plant provides an organic carbon derived from photosynthesis, and the bacteria is producing fixed nitrogen. Now, if you think about nodulation, you can already think that the plant has to now first allow the bacteria to get in. The bacteria has to form a kind of a home for itself through nodulation. And, it becomes kind of some endosymbiont inside the plant root. These are many, many steps in such a process. And this is a very complex process in which a lot of research is going on how is it that the bacteria is able to enter, how is it that it's forming these nodules, and plant is allowing such a bacteria to live in it for long term. So we still don't have all the answers, but we'll just go through what is the basics. Yeah. So as I told, it's an exchange of nutrients. The plant produces organic carbon, which is required for this bacteria, which is living in this, this is the bacteria. It fixes nitrogen to ammonia and it is exported to the plant cell. Now the plant will assimilate this ammonia and use it for its protein production or incorporate into amino acids. And this is what, we get. And this is also the basis of crop rotation, as if you remember that leguminous plants are always used in crop rotation in agriculture to enhance the nutrient status of the soil because they are able to fix this nitrogen. But not all plants are able to fix nitrogen, and it is only leguminous plants which can do it. And why is it that we try to understand why it is like that? So the rhizobial legume biosymbiosis is one of the most sophisticated symbiosis, and it is a more recent entry. So the AM symbiosis is a late, it was 400 million years ago. If you look at the time scale, the rhizobial symbiosis is maybe 60 million years ago. Yeah. So the nodule is a unique root organ designed to support this rhizobia and nitrogen fixation. And upon such an infection, the rhizobia differentiate and into bacterioids, and they live inside these nodules. And if you take a cross section, you can see this bacteria inside the nodules. So there are many labs in India and abroad who work on such rhizobial legume symbiosis, and it's still a hot topic. How is it that the rhizobia is able to infect these legumes? Now, what is the chemical dialogue? How do they talk to each other? We have seen that strigolactone and MIC factors. So in the case of such a leguminous plant, the plant roots produce specific flavonoids actually. Flavonoids are secondary metabolites and which are released from the root and which attract these bacteria. Yeah. So the bacteria knows now this plant is my friend. It is producing this flavonoid which is very useful for me. And now the rhizobia in turn, via the action of flavonoids, produce what is known as the node factors, that are those small molecules produced by the bacteria, which tells that it's symbiotic, and then the plant prepares it for a nodule formation. I mean, it's a very simplified model for this. Now, how is it that the flavonoids are activating these node factor production? So the plant flavonoids are known to stimulate actually the transcription of node genes in rhizobia. So these are the roots which are sending the small metabolites. They are taken up by the bacteria and they move into the bacterial cell, and it binds to these specific promoters and activate this node transcription factors to produce this nodulation, or the node factor genes are activated. And now once they have a node gene product, synthesize what is called as the node factors. The node factors is one of the earliest elicitor or such a small molecule which was discovered in any symbiosis. And it has a backbone also like the MIC factor and an acetylglucosamine or a chitin backbone, and with a lipid moiety which has a different decoration than the MIC factor. And this is being recognized in the legume symbiosis. Now, if you look up its recognition by the plants. Now, the plant produced flavonoids, the bacteria produced node factors. Now, how does the root initiate that good symbiotic signaling? Like the AM symbiosis, the calcium spiking in the roots is a key element even for node factor recognition. This is a root of a legume, and this paper has imaged how when you have node factors, when you add node factors from a bacteria into the root, what happens in the cell. So what you see here is you can see that there are, as you add node factors, you can see this red spiking. The red color indicates increased calcium, and you can see increased calcium in these frames and at a very specific place. So these are nucleus, and you can see this calcium increase only in this nucleus in the roots. Now, if you quantify these calcium responses, or what is red in color, you can see that these are like spikes that we saw also with the AM symbiosis, and also they are occurring in nucleus, which seems to be a nuclear calcium spike, seem to be central in both the symbiotic process. So upstream of these nuclear calcium spikes, the node factors or NF, what we see in this picture, and the MIC factors or the MF has to be sensed by the cell or the root cell using specific proteins. We call them receptor proteins. They are sitting on the plasma membrane and sensing these, bind to these ligands. These node factors are ligands which bind to these receptors and activate what you see is your signaling pathway for symbiosis, which involves downstream of it a calcium spiking. So these are one of the first proteins which interact with this node and MIC factor. Now, with respect to node factor, the identity of the receptors are well known. With MIC factors also, now recent reports have identified the plant receptors which are sensing these MIC factors. Yeah, so basically they are cell surface receptors. If you've heard my talk so far, you also know that both these pathways have a lot of commonalities. One commonality, they seem to have similar kinds of, chemical dialogue. This lipocyto-oligosaccharide, they can be different, kinds, but essentially it involves recognition of a node and MIC factor by receptors or channels which are upstream of this calcium spiking, which is common in both the interaction, and a signaling pathway which is common, and it's called a common symbiotic pathway. So whatever is marked in red are the SYM genes which are common between nodulation and mycorrhizal formation. So you have these receptors, channels, which is producing calcium spiking, the CCAM-K, which are calcium sensors, which decode these spikes and results in further, signaling pathway. These are all common for both the symbiosis, and hence point to the fact that they evolved from, or the AM symbiosis is the first symbiotic process from which the nodulation signaling pathway would have evolved. Now, the scientists are trying to incorporate this nitrogen fixing ability also into the cereal crops now. For example, in rice, you know that rice cannot accommodate a legume, a symbiotic nitrogen fixing bacteria, or it cannot nodulate. So scientists are trying to now, tinker with these common signaling pathways and trying to see if they can make rice fix nitrogen. Now, this is what we know about the common symbiotic pathways or the common interactions which are known. Now, both this AM symbiosis and leguminous, interaction with rhizobia are specific. As I told you, AM symbiosis is more spread out. 71% of plants interact with AM symbiosis. But the Brassicaceae plants, or plants like Arabidopsis or those present in mustard family, do not interact with AM symbiosis. So one such fungi which I work on is called Piriformospora indica, which is a mutualistic root endosymbiont, which is belonging to this order Basidiomycota. So what is very special about this endophytic fungus is that it colonizes roots and it promotes growth. It not only enhances nutrient acquisition, but it is also promoting growth. This is a pot without the fungal colonization, and this is after co-cultivation with this fungus. So you can see bigger plants when this fungus is colonizing. And they produce this pear-shaped spores, so they have this name Piriformospora indica. And one of the nicest thing about this fungus is that it can be grown without a root. Most of this AM symbiosis or these fungi which form symbiotic interaction cannot survive without a plant host. But P. indica can also be cultivated accinically, and it can colonize the roots of almost all the plants. There has never been a non-host for this fungi and promote growth in most of them. Now, surprisingly, this fungi was isolated in the Thar Desert by an Indian scientist named Ajit Verma. He isolated it from the rhizosphere of certain shrubs in the Thar Desert, in the microbiome of these shrubs, which were able to survive in the desert. So basically, he got this fungi from such a microbiome analysis, and which is what our lab has been working on. And I did my PhD on this fungi. So it's now, what do we know about this fungal colonizing the plant? Unlike AM symbiosis or rhizobial nodulation, there is no special structure that the fungi forms. In AM symbiosis, you have seen that they form arbuscules. In rhizobia, you have seen that they form nodules. But this symbiotic fungi doesn't form any of such, special structures. They are colonizing mainly in the rhizodermal root hairs and cortical cells, but they do not enter into the vasculature. And you can see the hyphae of the fungus growing on the roots, and this provide plants with multiple benefits: nutrient uptake, disease resistance, stress tolerance, etc. This is an example of a tomato root without the P. indica colonization. This is after 40 days after co-cultivation with the fungi, and you can see that the tomato root has really enhanced rooting in multiple plants. Now, what is the advantage with respect to nutrient acquisition and this fungus that I'm talking about? So, P. indica enhances actually phosphate and nitrate uptake in plants. This is an experiment which shows that this fungi has what is called as a phosphate transporter into them. Now, these are maize plants in this figure, and they have fed these maize plants with the radioactive P32. You can just a moment. Are you able to see my slides yet? Just a moment. I'll just stop sharing and start again. Yeah, I hope you're able to see my slides. So this is an example in maize where the P. indica is colonized, and with this where this PIPT gene in the P. indica is present, and what you see with the color is phosphate transport in the root and in the shoot. Now, when they knock out this gene in the fungi which transport phosphate, you no longer see phosphate acquisition. Yeah, this is with the wild type, with the normal P. indica, and this is when you knock out the phosphate transporter. And it also stimulates nitrate uptake. So these are reasons by which this fungi is able to enhance growth. Now, apart from producing nutrients for the plant, there is also a chemical communication between the plant root and the fungi in this case. So the P. indica, through its cell wall extract and culture filtrate, there is a active factor called cellotriose, which is present which is released by the fungi and which is being sensed by the plant root. We still do not know what is the signal that the root produces to accommodate this colonization, like flavonoids or strigolactones, what you saw in the other two interactions. Apart from the cellotriose, the fungus is also able to produce plant hormones on its own, even without the root. Then the fungus, when you grow it axonically, produces both auxin and cytokinin. So so there are multiple cues coming from the fungi, which helps the plant recognize it. And as I said to you before, this colonization results in enhanced nutrient uptake. It produces plant hormones and it gives stress tolerance, and hence it's very important. So now, what are the signaling pathways which operate upstream of this growth promotion response? Now, if you look up at the microbial signals which I have described so far, for example, I have told you about AM symbiosis, the rhizobia, and P. indica. Now, these are the three endosymbionts or symbionts that we are discussing today. The AM symbiosis, which is the oldest, which is one of the, or we'll start with the rhizobia. The rhizobia produces what is known as the rose knot factor, but its host range is very, very small. It only, it only colonizes the legumes. On the other hand, the AM symbiosis produces what is known as the MIC factor, but it has a little more advanced host range. Now look at the biomolecules which are produced, the P. indica. It is thought that it is a more ancient interaction because it absolutely has no host specificity. All the plants tested so far are able to be infected by it. And look at the biomolecule produced, it's similar to the MIC and node factor a bit, but it doesn't have these decorations which make these, AM fungi and rhizobium so specific. So this is what I just want to give you a hint that these microbial signals are also probably, responsible for its wide or narrow host range. Now, when we think about these microbial signals and how they are perceived by plants, it's also important to understand that the rhizosphere will have many kinds of microbes. So, microbial presence in the rhizosphere is detected by different plant receptors. The good, the bad, microbes, everything is detected. Now, the structural similarity between the node and MIC factors, that is, they are chitin fragments with decoration, and chitin as such is also being sensed by plants as being a pathogenic elicitor. This raises the question how these similar molecules could apparently induce opposite impact in plants. So one leads to a defense pathway. So if the chitin is being recognized by these specific receptors, it will lead to a plant immune response. A plant will defend itself against this pathogen. But if there is an AM symbiosis, it is able to accommodate it. So how does the symbiotic interaction evade this very very strong plant defense response in plants? This is a question which many people are trying to work on. And we are also, through this system of Piriformospora indica, trying to understand how is the microbial symbiont recognized by the root, how is the plant perception different from pathogens, and what are the key genes involved in this interaction. So as I told you before, with respect to the MIC factor and the node factor, that the calcium fluxes are key to recognizing it. Even in the case of P. indica, many years ago, we discovered that the P. indica, cell wall extract, which contains this active factor called cellotriose, is able to activate a very rapid calcium elevation in the cytosol. Yeah, so this is one of the hallmarks of its recognition by the roots. So these calcium elevation are very specific to the roots. If you put this cellotriose in the leaf, you will not get it. So it's only in the roots that you get these specific calcium elevation, which activates a signaling pathway. Now, since these calcium elevations are crucial in symbiotic interactions, as I told you before, they are upstream of all this pathway. So we wanted to know what are those channels. There are certain proteins on the plasma membrane or the organelles which are responsible for producing this calcium spike, or they bring the calcium in from where there is excess amount of calcium. So we were trying to look at those calcium channels which are important for such a calcium elevation with respect to P. indica and Arabidopsis. So we looked up at we did a microarray, that is a gene expression analysis, and looked up at those genes which were activated very rapidly when you have this cell wall extract or the cellotriose treatment. And we figured out that this specific protein, the CNGC19, is highly induced when you add the cell wall extract, but not the cellotriose. And we decided to look what is the function of this protein. So you can see it's highly induced when you treat it, and it's also induced when the fungus colonizes the plant. Now what we did, we had mutants which do not have. If you have to find a function for a gene, you will have to knock out that gene or suppress it in some way and see what happens in the plant if you don't have this gene. Yeah, so this is what you call a functional assay or functional characterization. So we do a plate growth assay for P. indica treatment in Arabidopsis. This is a control plant which has no treatment with this special fungi. Now, this is a P. indica treated plant for the same time points, both of them. And you can see if you treat it with the fungi, you see that the roots are more branching and there is enhanced, both root and shoot biomass in Arabidopsis plants. Now, this is what happens if you do not have this channel or the CNGC19 channel. This is the control plant, and this is after co-cultivation. So if you can see that after co-cultivation, the plant seems to perform worse. Not only does not promote growth, it also has kind of become a little sick. Yeah, we also see this phenotype on soil. So this is in soil, this is the wild type plants, this is without the fungal treatment, and this is with the P. indica treatment. You can see that the plants have grown bigger. But if you do not have this channel, which is responsible for at least part of the calcium elevation, you no longer see this enhanced shoot growth with both the mutants. So there is not only no growth promotion when you don't have this channel, but there is also growth inhibition when you do not have this channel. This is what we quantified also here. Now we try to ask ourselves, why is it that if you don't have this channel, the plant is, instead of promoting growth, it seems to repress growth? So we looked up, we have some P. indica, which is GFP labeled, and we looked up at what happens in the wild type plant when the P. indica is colonizing normally, and what happens in this mutant, which does not have this calcium channel. So it's very obvious from this picture that in the wild type, the fungus seems to colonize in a balanced manner. There is not too much colonization, yes, but you see some colonization. But with respect to when there is no this calcium channel, you can see that the roots are heavily colonized. So the P. indica colonization is enhanced in this loss of function mutant, and this the plant doesn't tolerate. The plant somehow tries to control the colonization of fungus. It doesn't mean that it's a good fungus, so my roots have to be fully covered with them. The plant tries to regulate the colonization in which there is an optimal balance of the fungal colonization, which is lost in the mutant. We also did the same experiments. We quantified them with real-time PCR, and you can see that in the mutant, there is enhanced colonization, and which shift the balance from mutualism towards pathogenicity. Now, apart from this response, we also asked ourselves what happens to this elevation that we saw with the cell wall extract. So this is, that is, I showed you before with the NOT factor, the MIG factor, and also with P. indica that there is a calcium elevation. Now, if you do not have this channel, this specific channel, even this calcium elevation seems to be reduced. It's not the only one responsible, but it is involved in this elevation of calcium. And hence, it's important for the symbiotic signaling. But the cellulotriose dependent pathway is completely untouched by the CNGC19. Now it is known that, the plant immunity is very, very crucial for P. indica colonization, and we call it a MAM-triggered immunity. This is the basal immunity in the plants when it encounters any microbe, good, bad microbe. Yeah, so we know that this MAM-triggered immunity is critical for controlling the root colonization by P. indica, even though it is a very good microbe. And this balanced colonization in certain specific regions in root is maintained by this very robust MAM-triggered immunity. And one of the determinants of it is a secondary metabolite called indole glucosinolate, which participate in this MTI. There are multiple plant hormones, glucosinolates, and defense genes which are involved in this process. And we went on to ask what happens to this immunity when you don't have this channel, and why is it that it's colonizing so much? So we found that most of the defense genes which are activated during P. indica colonization, which are GA marker genes, some of them involved in SA pathway, they are usually upregulated in the wild type. So the plant is able to sense this microbe, though it is a good microbe. It is not like that, it is activating some basic defense. So you can see this black bars, they're all activated usually in a wild type. But in this mutant, you have seen that the plant lets its guard down and it no longer recognizes it as, there is no active defense. And this results in an impaired defense gene activation and an enhanced colonization, which is also not good for the plant. We went on to look at other markers. As I told you, indole glucosinolate is a secondary metabolite which we can quantify by HPLC. In usual way, in wild type, which are the black bars, you can see upon colonization of this fungi, you can see an increase in the indole glucosinolate content, which is lost in this mutant. So, the indole glucosinolate levels are also not regulating here. So, so this is also one of the reasons why the fungus colonizes more on this mutant than in the wild type. So based on all this study and many other data, we were able to, come together at this theory that the P. indica is colonizing this, the roots of Arabidopsis via, unknown elicitors. It is not cellotriose in this case, and we still don't know what receptors. As I told you, in the case of node and MIC factor, we know that there are certain surface receptors. We don't know here what are these receptors, but there are some receptors which sense it, which in turn activates this specific calcium channel, which is responsible for this calcium elevation, which is a hallmark of most of the symbiotic interactions. And this also activates, in this case, a very active plant immunity, which seems to control this P. indica colonization. Now, if you lose this channel and this MAM-triggered immunity, you will get excess colonization. So well-defended plants are not only good for pathogenic interaction, they are also critical for mutualistic interaction, as in the case of Piriformospora indica. This is what we published this paper last year. Now, what is known in other systems? There are certain systems like rice. Now, if I am coming to this concept of pathogenic fungi or differences in the perception of pathogenic fungi and the AM symbiotic fungi in this case, how do they recognize something as being a symbiont and an immunity signal? So different systems have different, different mechanisms of how this is done. This is the latest paper which came last month, this month in PNAS, where they have tried to understand how symbiosis and immunity signals are discriminated by receptors. I told you there are certain receptors on the plant surface, at least in case of Arbuscular mycorrhiza. So these are Arbuscular mycorrhiza, which is sending out this chitotetraose, which is its elicitor. Yeah, it is being sensed by this specific receptor, which is called the OS-MIR1, which activates the symbiotic signaling. Yeah. And now, how does we know that it activates symbiotic signaling? We also know that it is the same chitin fragments which are being released by pathogenic fungi and which activates, which activates defense response. However, the receptors in this case are different. The receptors which sense pathogenic fungi are OS-SERC and OS, these green colored guys are the one which sense the pathogenic fungi, and the orange colored protein is what senses this Arbuscular mycorrhizal signal. Now, when you come to the slide where you see a part where you are explaining the Arbuscular mycorrhizal part, what this OS-MIR1, which is a receptor of these MIC factors, does is that it depletes this, there are two proteins which are involved in sensing the pathogenic fungi here, as you can see here. He depletes one of it. He inhibits the, prevents this complex formation from happening, and hence it cannot recognize these signals. So the OS-MIR1 initiates both symbiotic signaling and represses rice immunity by depleting the receptor-like combination kinase OS-SERC1, preventing the formation of an immunity complex. So this is one of another mechanism by which plant is able to sense a pathogenic and a symbiotic fungi. So I'm just, this is my last slide. And from this talk, this is the take-home lessons which I want you to think about, that the microbial superheroes, as you know from my talk, it enhances plant nutrition. There is a chemical communication between plant roots and microbe, which determines the symbiotic interactions and are unique for each interaction. For example, the chitin in fungal cell wall, which are beta-1,4 link polymer of N-acetyl glucosamine. In its unmodified form, they are strong inducers of plant immune response, but when they are acylated, they act as symbiotic factors. Yeah, so this chemical communication is essential. There is a location-specific calcium spikes which are crucial for a plant to recognize a microbe as symbiont, and plant defenses are very much active during symbiosis, and depending on the kind of symbiotic interaction, various strategies are used by plants to balance this, as I told with P. indica, or as I told with this example of rice. This, I want to come to the end of my talk. This is our group in NIPGR. We are mainly working on plant-insect interaction, but we also have few people who are working on this Piriformospora indica response. We are funded by DBT, ICR, and EMBO, and the Max Planck Society. You can follow us, our work on our webpage and also on our Twitter account. So thank you very much. I'd be happy to take questions.
Thank you, Jyoti, for the fantastic talk. There are some questions posted in the Q&A box, so I'll just read it out, then you can answer them. So Atar Ambedkar has a question. What methodology is used for quantitative estimation of calcium spiking in nucleus?
So we use genetic reporters or transgenic plants which are expressing these calcium reporters, and various kinds of these reporters are available. They bind to calcium and release a light signal, basically. See, in most of this slides, what we have shown is either an ecorine based calcium reporter, or a chameleon based or GCAM based. There are different calcium reporters which are transgenic plants, or many cases also in animal system we use dyes, but in plant system they are transgenic plants which sense this calcium elevation and give us a readout in respect to certain light signals which we use to quantify. And you can also image these plants as it happens when you add a signal, and you can image this calcium elevation inside the plant cells.
Okay, thank you. So Rudra Banerjee has two questions, very interesting ones. First is whether host microbial association varies spatio-temporally. And the second part is what factors determine localization of fungal hyphae inside the host plant?
Yeah, so spatio-temporally, yes, different microbial interactions, as you have seen with the well-known, MIC and the node factor interactions. These are only at specific zones of the root. Also with P. indica, it's not that you see them colonizing all the cell types in the root. So the plant is also able to control this colonization in specific part. We still do not know how do they do it. How is it that they keep them away from the vasculature, for example, which is a highway for it to transport all the nutrients. They seem to keep it till the cortex. We are still studying how is it that they do it. But we know that many of these proteins which are involved in this interaction, all these calcium channels, for example, they are localized to specific region in the roots. And this could be one of the reasons why we see the spatio-temporal regulation.
Okay, so there is a question from Srinidhi. Whether you can suggest any books on plant microbial interaction that they can read later.
I would suggest nice videos, you should in YouTube there are very nice videos on nodulation from Sharon Long in Stanford. She was the one who discovered these node factors and their interaction. If you have a chance, please go through these videos from Sharon Long, and iBiology series, which I absolutely love. But other than that, of course, there are a lot of literature. I cannot tell any specific books, but yeah, there is a lot of papers which you can use. Maybe you can write to me and I can send you a few.
Yeah, hope it answers your question. So there is another person who has asked, Viraj Vishnu Prasad, whether all microorganisms produce node factors and can form root nodules.
No. It's only this rhizobial bacteria which produce node factor, and it can be only recognized by legumes. This is what I'm telling you. So nodulation doesn't happen in rice. It happens only in that specific genus of plants. So that is what scientists are trying to now, see why is it, that the node factor is not being recognized in other plants. So there are certain receptors in the plant surface which are doing it, and which have obviously is different in different plants.
Thank you. So Atar has raised another question, that whether is there a possibility that if there is an evolution of a pathogenic fungi that can mimic signaling factors to evade plant immunity and establish infection?
Yes, exactly. So I think the MAM-triggered immunity or the pathogen, sensing mechanism is what evolved first. All these symbiotic, processes may have evolved from this basic MAM-triggered immunity. This is my feeling about it. So evading this microbe is the basis of evolution of most of the symbiotic processes. But the role of plant defense in symbiotic interaction, it is coming up more recently because people have always focused on what are those genes which are responsible for symbiotic interaction. They have never really thought that there could be a very active plant defense which is involved in this process. So a lot of studies are coming up which are telling that there is somehow an active suppression of immunity, or somehow in some cases an immunity is required, as in the case of P. indica, where immunity seems to be critical for controlled colonization. In case of rice, as I told you, in the receptor itself, the symbiotic receptor tries to stop that immunity receptor from forming a complex. So different plants have different systems, and many of them are waiting to be discovered.
Thank you. So maybe the next question is can be answered together with this one, like whether how do plant defenses control and keep the fungus from keep them from wiping out the fungus in healthy amounts or something. I think what the attendee meant is, yeah, I think you got it. Yeah.
How does they maintain that it's not too much colonized?
Exactly.
Plants produce a lot of, signaling components along the way. So, for example, ROS is a very important determinant which is involved in plant immunity, hormones. So, different things which prevent the fungi from colonizing too much. They are all part of plants, kind of an antibody which makes it not colonized too much. So all these are already present in the plants. There are a lot of pathogenic microbes around, but the plants are all green still. So the plant immunity is very robust, and he doesn't let the plant immunity doesn't let these pathogenic fungi to colonize too much in anyhow, using all these signaling components which inhibit their colonization or hyphal branching at some stage.
Yeah. Fantastic. So I'll combine two questions, which is about the MIC factor. So the first one is whether strigolactone and MIC factors are species specific. And the second question is how do MIC factors stimulate root branching?
Okay, so I really don't know how MIC factors regulate root branching. A lot of studies on it has not been done. MIC factor is just isolated in 2011, according to me. So what was the first question, Radhika?
Whether MIC factors and strigolactone species specific.
They are specific. So at least the MIC factor is very host specific, and there are different kinds of this, backbone and that decoration of the fatty acid. They seem to be different in different. For example, Medicago will have one type of it, Lotus some another kind. So they are really host specific.
Okay, Dr. Yogesh has a question, whether very specific to bamboo system. So he's asking, how can we enhance the root with the help of growth hormones in bamboo to get a huge diameter?
You can try with P. indica. It doesn't seem to have any host specificity. And if you are looking for a non-transgenic method to enhance plant growth, I mean, this could be one of the techniques you can try. Whether they colonize the roots of bamboo, I don't know. But just adding hormones is not going to give you more growth. I mean, how much hormones will you add? So, but the fungus or live fungus can give much more advantage. And if they don't have a species specificity, what more do you need? So it can be tried. I mean, these, the bamboo will also have its own microbiome, which will have different collections of microbes which are good for bamboo to grow. So this is another way you can, explore how to enhance growth in bamboo.
So Sujin Bimal has raised a question, whether have you checked protein levels in response to calcium spiking after fungal attack?
What do you mean by protein levels after calcium spiking? So the calcium spikes in plant cell are being recognized by specific proteins which go and, this calcium go and bind to specific proteins inside the plant cell, which, activate a signaling pathway, which can be a symbiotic signaling pathway or a pathogenic or whatever, different signaling pathway. So proteins are essential for this calcium spike to be translated to something meaningful. Yeah, the calcium spike as such don't have any meaning. Only when they are transduced into, taken over by proteins, do they have a meaning.
So Anshika has a similar question as to, can we identify channel protein agonists for controlling the calcium spike? Is there a way to do this?
Yeah, in all these interactions that I told about, also in the micronodulation, many channels are known which are critical for these calcium spikes. So if you do not have them, or if you introduce them into a new plant, it might be. But I don't think just transferring a single gene is going to help the whole process, because as you see, it's a very complex process which has multiple steps where a microbe has to be going in, activating these calcium spikes. It has to be activating specific set of genes. So of course you can do it, but you need to have a holistic idea of the whole pathway.
Thank you, Jyoti. There is one, I don't know, it's a student, but Shruti Mishra has raised her hand. So Shruti, can you please ask your doubts?
Shruti, just unmute yourself if you can. Or else, please put your question in the Q&A box.
No, sorry, that was a mistake. I raised the hand by mistake. Sorry.
Okay.
What about Ashwin? Ashwin, please go ahead.
Hello, ma'am. Am I audible?
Yeah, I'm.
Ma'am, my question was, how does a plant decide when a when a fungus colonizes a plant? How does a plant decide that this much is enough? Or since immune systems are activated, how does the plant how does a plant prevent itself from killing fungi which are in healthy population numbers?
Mm-hmm. So there will be multiple processes which are responsible for it, that it doesn't kill the good fungi. One of it is you have to start from the very beginning. What is perceived? Now it perceived, for example, a node factor. It already has receptors which are very specific proteins which specifically bind to these ligands, and it already knows it doesn't have to activate too much of a defense pathway, because there are a specific set of proteins which are binding to such ligands. So that is the first step in recognition. Now, downstream of it, as you saw, that there are calcium elevations which are produced. These calcium elevations are very different for different microbes. So some happen in the nucleus, some happen in the cytosol, some have spikes, some are just one, big elevation. So these also help it to determine, or such signatures of calcium also help it to determine whether it is a good or bad microbe. There are also step 3, the pathogenic microbes, or both of them. I think both the symbiotic and the pathogenic microbes always have to overcome a basic plant immunity. But if they are not able to overcome it, the pathogenic fungus, for example, produce what is known as the effector proteins, which it injects into the plant cell, and which down-regulates this plant defense. In case of symbiotic interactions, the basal defense is, as I told in the rice interactions or in Piri, the basal immunity, after a certain time, the plant is able to make out that this symbiotic organism is not able to save, it might not produce effectors, or it doesn't activate too much of bad signals inside them and let them in. So the way they both are very different, but keep in mind that the basal immunity is critical to regulate their colonization from the respect of a plant. Now we don't know with the microbes what regulates this colonization. That is completely an unexplored area.
Thank you, Jyoti. There's one very interesting question that has come up from Sajeevan. So he's asking, will pathogenic microbes have advanced mechanism to bypass all immune response of plants and establish a disease, or will they have some sort of mechanism so that they mimic as the symbiont and enter the plant and then become pathogenic?
Okay, so that's a very nice question. I do not know of an example where they mimic a symbiont and enter into a system, but pathogenic systems have very well evolved to overcome this basal immunity. As I told you, they have specialized proteins called effector proteins which go and block all these activated pathways. So, for example, there are proteins which block the jasmonate pathway, which is activated when there is defense or the secondary metabolite. So these effectors are critical in for pathogen to infect. I do not know of an example where they have come up as a symbiont and then become pathogenic at certain stages. The other example I can think is of coronatine. So, for example, bacteria, when they colonize plants, they release, we know that jasmonic acid is very important for plant defense against insects, and salicylic acid is very important for plant defense against microbes. And they are very mutually antagonistic. So if JA is activated, the SA is downregulated. I know that bacteria, for example, releases a mimic of this jasmonic acid isoleucine called coronatine to suppress the SA pathway so that it can gain entry into the plant cell. Yeah, this is known, but not of a symbiont, some microbe acting as a symbiont and getting becoming a pathogen later. I haven't seen any.
I also don't know, but I think there are many epiphytic microbes that are harmless and then suddenly become pathogenic, right? So we don't call them symbionts, but epiphytes that are harmless and then suddenly they become pathogenic after some trigger from the plant. So one last question, I think, from Rudra again. So how this interaction would respond against an environmental gradient? He's asking whether this will change across geographic locations.
You mean the well-known symbiotic interaction of the rhizo or the microbiome? The microbiome might change, but I haven't come across, or I'm not aware of studies where they have looked at different gradients and how these interactions are modulated by it. I'm not aware of it.
So Dr. Saudamini has a question. So Saudamini, can you ask your question or should I?
Yeah, I could. Yeah, thanks, Radhika. Hi, Jyoti, very, very interesting talk and set of questions and answers. My question is more structural. Oh, you talked about how the chemicals are being given away by plants and are recognized by the microbes to say that this is a good host and so on. And one of them was decoration of chitin, right? So I wanted to know if we know the enzyme that is catalyzing the attachment of the lipid to the chitin. Do we know which protein, which enzyme is involved? No?
No, I do not. I'm not aware of it.
Okay. So, yeah, if it is a...
Again, combination of both node and MIC factor of how they bind is very well worked out. But with respect to more the bacterial part, I'm not aware.
Okay. Yeah, because I was thinking if it is a very bad pathogen, fungal pathogen, which is going to destroy the plant, we could address it at the root of the problem, as it were.
Yes, these signals are really at the root of the, problem. It really discriminates them being pathogenic.
Right. Yeah, great. Thank you.
Thank you. So I have one question, Jyoti. I hope I can ask one last one. So your CNGC19 mutant, you showed that at 42 dpi, the number of piri had reduced from 14 dpi, isn't it? That's in the mutant, the amount of piri infection reduced after some time point?
Mutant, the piri infection is very high. I mean, it over colonizes this root.
Yeah, that was at 14 dpi, but when you monitored it
on the soil, what happens that the indole, are you talking about that indole glucosinolate which goes down after 42 dpi?
No, I think you measured the piri colonies, or was it that graph? I don't know. There was 2 time points, 14 and 42. Was it wild type versus CNGC19?
Yes.
So is it like after some time, somehow the numbers come down? No, it doesn't. So the number is maintained.
And in different host plants, the speed of the colonization also seems to be different. For example, in tomato, it seems to need 40 days for that nice growth promotion, whereas in piri, it just needs lesser time. So
is there any phenotypic, like piri causes any symptoms at all, like infection or in these mutants, or there's no effect of?
Phenotypically, you don't see any. I mean, other than that, the plants are very sick when you co-cultivate them. And then when we measured all the hormones, all the defense pathways seem to be down-regulated.
Okay. Thank you so much for the very interesting talk and introducing us to the world of plant microbes. Now I'll hand over to Pavitra to conclude the session.
Hi, thank you so much, everyone. And 1 second, sorry, the chat is open. So thank you so much, everyone, for attending the sessions and interacting with us and asking so many thoughtful questions. The only thing I would like to add is also thank you, Dr. Jyoti. I think we all learned a lot about, the different kinds of symbiotic aspects. And I've put in for anybody who does want a certificate of attendance, please do fill in the form that's there in the chat. Okay, and it's only if you've attended all 3 talks, otherwise it's not applicable. Okay, please don't email us about it. And lastly, I would like to add a big thank you to Dr. Radhika for hosting these and for moderating and also sharing so much of her expertise in these areas. So thank you, everybody. We hope you've had a good time listening to these sessions, and you can watch them on YouTube on our channel. Please go check them out and share them with your friends. Thanks, everyone. Have a great day.
Radhika,
Bye.
Thank you, Jyoti. Bye. Thank you, Sadamini. Bye-bye. Bye, Pavitra.